high concentrations outside of cells. So when a calcium channel opens, it causes calcium to come into the cell, right? So what happens to calcium when it comes in? Think back to neurons. It's the same exact process. Calcium binds to regulatory proteins on these little synaptic vesicles. And when it binds to these regulatory proteins, it causes these synaptic vesicles to undock from an area called the trigger zone for them to merge and actually form with the wall or the membrane of this inner segment, just like how this occurred with the terminal buds of a neuron. So when calcium binds to these little synaptic vesicles, or neurotransmitter vesicles, whatever you want to call them, it causes those vesicles to merge their membrane with that of the inner segment membrane. So calcium floods into the cell and binds to these little vesicles, allowing the little neurotransmitter inside of there to diffuse into the synaptic cleft from high concentration to low, right? So glutamate will diffuse out. So this is kind of odd because this is the dark phase. So we're actually having the release of glutamate during the dark phase of your photoreceptors when there is no light hitting them. So when you close your eyes and no light's hitting your photoreceptors, cyclic GMP concentrations will increase, allowing sodium to come into your cells, depolarizing the membrane, allowing calcium to come into the inner segment, allowing calcium concentrations to increase in the cell, bind to regulatory proteins on these little synaptic vesicles, and glutamate to be released into a synaptic cleft. This happens every time you close your eyes. Most of the time for split seconds when you're blinking, so not a whole lot's really accomplished there. Here's my reason. Okay. So let's give me a little bit more room here. Now we should know from our discussion of photoreceptor anatomy that these photoreceptors synapse with the cell we call a bipolar cell, right? Which is just an upstream neuron that communicates signals to the brain, or 8-farad sensory neuron that helps carry signals to the brain. It's going to pass an action potential on to a ganglion cell, which will then form with other axons to form the optic nerve, carry the signal, vision signals to the primary vision centers in the occipital lobe of your brain. So we know that this bipolar cell, this is a real neuron. So it doesn't produce receptor potentials, it produces generator potentials. And we know that generator potentials generate action potentials, right? And carry signals to the brain. Now, we have little proteins studying the membrane of this dendrite of your bipolar cell that have glutamate receptors. So when glutamate's released into that cleft, that little synaptic cleft, it'll bind to these little tiny receptors. Now this is where things get kind of confusing, because in the way we've always learned neurons in the past, when a ligand binds to a receptor, it typically is going to cause one of two things to happen. Potassium to flood out of a cell and the neuron to be deactivated because of hyperpolarization or hyperpolarization. Or that little ligand to bind to a sodium channel and allow sodium to come into the cell and generate an action potential. Now these bipolar cells are unique though from other types of neurons, because they have sodium ligand-gated channels that are always open. always wants to naturally flow into the cell, creating a generator potential to generate an action potential. However, these little enzymes, these little proteins here that are found that bind to glutamate, create a product that closes these channels. So basically, in the dark phase of vision, so I guess it's the dark phase of non-vision, you don't see anything, these sodium channels are closed. And thus, these bipolar cells are always at normal resting membrane potential. So they're not generating a generator potential that can generate an action potential. So no signal can be sent to your brain that you're seeing anything. And so what does your brain perceive that you're seeing when the sodium channel is closed? Absolutely nothing, right? That's why this is the dark phase of vision. Now, the confusing part about this is that it's almost exactly the opposite of what you would think would be happening. You would think that, oh, well, this would trigger these, and that would open, and then you would get a signal. But no, it's exactly the opposite. You get high concentrations of cyclic GMP during the dark phase, opening these sodium channels, causing membrane depolarization and calcium influx, calcium binding to neurotransmitter vesicles, allowing glutamate to be released in the cleft, glutamate binding to enzymes that help catalyze a product that actually closes these sodium gated channels, which thus prevents any signal from reaching your brain, which is why this is the dark phase of vision. Make sense? All right, so what happens in the light phase? Let's draw some more photoreceptors. Process again, but now we're going to do the light phase. Put our little opsin proteins on our discs in the outer segment again. Okay. So what happens in the light phase? Well, number one, we need a light wave. So here's a light wave. Well, actually, light waves will be coming from this direction. Here's a light wave. Hitting a little rhodopsin protein. So these little discs, we'll just call all these opsin proteins rhodopsin just because it's easier. So rhodopsin protein. So your little rhodopsin proteins are interesting because they're the ones that actually interact with light. They're the ones actually stimulated by light. And not unlike a voltage-gated channel where you have a voltage change and redistribution of electrons and opening of a channel from proteins changing shape, these rhodopsin proteins change shape when they're stimulated by light waves. Light waves hit them, cause them to change shape, cause them to change their form. So when rhodopsin's hit by light, it changes into its activated form, which we call metarhodopsin, all right? Which is the first stage of light perception. When metarhodopsin is formed, it's actually interacts with, let me move this arrow a little bit. Each of these little rhodopsin proteins interacts with something that we call a G protein complex. We really don't go over G proteins a lot in anatomy and physiology, but in case you did in biology, you kind of know what I mean. But it's a G protein complex that we call transducin. We'll draw it as a diamond. I don't know why. Transducin. And basically, transducin is going to help transduce this light signal into a chemical form. All right? So when a light wave hits rhodopsin, it causes rhodopsin to turn into meta-rhodopsin. At the same time, that shape change of rhodopsin activates transducin. So this transducin G protein complex is activated by the shape change of rhodopsin into meta-rhodopsin. Now those of you who learned G proteins in the past know that that really means the G protein complex dissociates. But again, we're not interested in going into that detail, so we're going to go with activated. Sounds good to me. Activated transducin. Now, transducin has a particular function that's quite interesting in terms of how this all works. Color. Transducin actually activates, so we'll do a little positive there, activates a protein called oh, I forget what the actual term is. It's one of two different things. This is my notes. Yeah, phosphodiesterase. It's cyclic GMP phosphodiesterase. Right? So transducin activates when it's activated by rhodopsin changing shape to meta-rhodopsin, activates a cyclic GMP phosphodiesterase. This is important because remember, our cell nucleus is constantly producing enzymes that's creating cyclic GMP, which is our ligand to those sodium chains. However, when transducin activates cyclic GMP phosphodiesterase, this phosphodiesterase basically takes cyclic GMP and breaks it into little bits, thus destroying any functional capability of that molecule. So cyclic GMP phosphodiesterase destroys cyclic GMP. It basically catalyzes the breakdown of that molecule into simpler molecules. Now you might remember from A&P2 that when we have channels and ligands, ligands are typically very specific for a specific receptor in order to open up a channel or to stimulate some type of enzyme. So when cyclic GMP starts getting destroyed, what happens to our sodium channels? Instead of being open, they're closed, right? Why? Because we have no cyclic GMP. Because cyclic GMP is rapidly being destroyed by cyclic GMP phosphodiesterase, which was activated by transducin, which was activated by rhodopsin changing shape to meta-rhodopsin, which was activated or stimulated by a light wave hitting it. So, if we drew our little graph again, here's our resting membrane potential. So RMP over time. If we have no light